Hello, I'm Dr. Marc Rothenberg, the Director of the Division of Allergy and Immunology here at Cincinnati Children's Hospital. I'm also the Director of the Cincinnati Center for Eosinophil Diseases, which is an internationally recognized leading center whose mission is to better understand, treat, and cure eosinophilic diseases. This talk today is on the occasion of National Eosinophil Awareness Week, which is a very meaningful, significant period of time established 18 years ago by the House of Representatives here in the United States in response to patient advocacy. This week brings attention to the United States and beyond on the importance of eosinophilic diseases. Awareness is raised, importance of patients' voice in dictating policy, as well as the importance of the attention of government officials and key advocacy and key stakeholders across the United States and beyond is drawn to all of our attentions. This week is particularly significant, and it shows the power that the individual patients can have through advocacy to influence policy and truly impact the course of this disease. This is especially timely in view of the substantial cuts that are taking place in federal funding in the United States currently, and the importance of making sure that we can sustain and, in fact, enhance the attention that these diseases deserve. What I'd like to do in this lecture today is share with you new insight into whether or not eosinophils are actually the causal cell of this set of diseases, eosinophil-associated GI diseases. I will particularly focus on eosinophilic esophagitis as a main focus area and topic. And the question that I will address is, are eosinophils the right target? Meaning, should we be focusing on these cells as the causal cells, and hence the importance of eradicating them to improve the patients with these diseases? Now, this attention is drawn and deciphered through the work of my laboratory, which is focused on understanding and treating eosinophilic esophagitis. My lab has several different components. In one arm, there's the fundamental research studies, and the other arm is genetic studies, where we try to understand the basis for heritability of this disease. We also have an arm of the lab, which is translational medicine, where we can model eosinophilic esophagitis in vitro as well as in vivo in animal models of the disease. Now, if these disciplines reinforce itself, then we can go and test their theories in patients through clinical studies. And in fact, we've been able to advance findings from the fundamental lab and genetic leads that we had into positive results in the preclinical translational systems, and then move them into proof of concept studies in humans, and then ultimately into drugs that are now approved and others that I'll discuss that are in the pipeline for better treatment and hopefully curing eosinophilic esophagitis. Now, the focus on eosinophils itself started quite a number of years ago, when I spent a good part of a year considering the research that we had just begun to uncover and trying to understand the nature of these diseases. And I coined the term eosinophilic gastrointestinal diseases, which is known by the acronym EGID. In this simple diagram, I placed EGID in the middle of the spectrum between pure IgE-mediated diseases and those that not IgE-mediated. IgE-mediated would be like anaphylaxis to peanut. On the other hand, with non-IgE-mediated immune diseases that are purely non-IgE-mediated is one like celiac disease or gluten allergy. EGID falls in the middle category. It has properties of both. And the specific type of EGID we defined by the region of the GI tract that was involved And what's shown here is the different designations of what we called these diseases back then, and there's been some modifications to this more recently. One of the great advances that science has given us the capacity to do now is to remove eosinophils from people. And this is something that I refer to as an eosinophil knockout, a human eosinophil knockout. This means that we are selectively removing eosinophils by specific drugs, particularly antibodies, that will deplete them in a very safe manner. And this article reviews our experience with this. And we brought together experts from around the world to come together. And one of our conclusions was that eosinophil knockouts in humans are safe, so that blocking allergic reactions is not very detrimental. And in fact, we believe that eosinophils, through this set of data, are vestigial, meaning that they do not have major consequences on the host immune response. They're not associated with protection against common germs. But this provided us with an opportunity to ask the question, are eosinophils causal in eosinophilic esophagitis? And I won't go into all the data that's emerged, but want to share with you this result here, which was a late-stage, multiple-site, double-blind, placebo-controlled study looking at a particular drug called benralizumab. And we used this drug to remove eosinophils from the body and then see what happens to patients with eosinophilic esophagitis. Now, in part A, you can see that the drug induced a removal of eosinophils called the histological response in nearly all the patients, in this case 87%, where the placebo was less than 10%. So the drug worked. In fact, many of these patients had 0 eosinophils left in their body. Now, despite this, what's shown in B, that the symptom scores, using something called the Esophageal Symptom Questionnaire, but that's not that important for the basic take-home message, is that symptoms did not improve compared to the placebo. Other features of the disease, such as the endoscopy and other markers, also didn't improve. So these results really changed us a lot in terms of how we think about this disease, eosinophilic esophagitis. Although we can say that esophageal eosinophils are an excellent biomarker for diagnosis, they are not likely to be the causal cells in terms of eliciting symptoms and other clinical disease features. Now, they may be useful as a biomarker for monitoring disease activity under some conditions. So now the question comes up, which is the heart of my talk. If it's not eosinophils, what are the causal cells and disease mechanisms? So the first thing I want to point your attention to is that EOE is an allergen-induced immune response. And we showed this 25 years ago when we first started working on this. We thought that esophagitis was mainly provoked by acid exposure, but in this case, it's just an illustration of a large amount of data. We took animals and we challenged them with allergen. And what we found was that eosinophils would accumulate in the esophagus following the allergen exposure. This is the controlled saline, and this is the allergen here. And you can see very large levels of eosinophils that accumulate in the esophagus when we intratracheally or intranasally challenge them with an allergen. So this really changed the way we thought about this response, because it indicated that this is related to allergies and not acid. We can see this in humans also, and that's illustrated in this figure. On the left, we see a biopsy from patients with active eosinophilic esophagitis. These are the eosinophils, these small cells here that are red staining. This is the squamous epithelium in the esophagus that's actually showing a lot of changes. When we remove foods, food allergen elimination, we can restore a normal esophagus. We don't see a single eosinophil. This is a reversible response, which is very encouraging for patients. But we know that if we re-expose patients to foods, they will relapse. We can also, as I said, do this in animals. Now, we know that this is a complex process because the food allergens that one avoids isn't necessarily that easy to identify. but work done by the Consortium of Eosinophil GI Disease Researchers, which is a group of outstanding physicians at 16 different institutions in the United States, working together with conjunction with the patient advocacy groups, Cured and ABFED and others, that we showed in this study that food elimination is effective, but also that milk elimination alone can be effective as well. In fact, removing 6 foods and 1 food, milk, was equally effective. And this data was summarized in a press release by the National Institute of Health, which is summarized here, indicating that milk elimination is an effective early treatment for eosinophilic esophagitis, and it does work, and even may work as well as excluding 6 different foods. The next message that I want to bring to your attention is that EOE is a hereditary disease involving complex genetics and environmental factors. And we found this out in the original studies that we conducted with twins with EOE. And what you can see here is that monozygotic twins, basically identical twins like these 2 babies here, have a 40% concordance, meaning that if one twin has EOE, the other one has a 40% risk for the disease. Now, that's a large risk, but it's not 100%, which you would see in a purely genetic disease. It indicates that genetics is important, but the environment is also playing a critical role. And in fact, when we looked at fraternal twins, or we call that dizygotic twins, we saw also a high concordance, which was about 10 times the rate that we see in regular siblings that have the same amount of DNA sharing, but a different early life exposure, like in utero and in the first year of life. So this indicates that genetics is important and that the environment is interacting with the genetics. And in fact, a number of investigators are working on what those environmental factors are, which is certainly things that are important in early life exposures that predispose to EOE. In terms of the genetics, this is an example of some of the genetic leads that we now know that are important. This is a complicated slide, but what we're looking at is all the regions in DNA that are coded for different chromosomes. And these dots represent different patients. In this case, we're looking at 1,000 different patients with 10,000 different controls. And we're looking at here is different regions in the genome. And these regions in the genome have a susceptibility over here and over here. This is what you call a Manhattan plot. And it shows that these particular regions of DNA genetic variants predispose to the disease. And some of these are quite dramatic in terms of the magnitude of the statistical significance. But one of the main points of these findings is that nearly all of these genes are not eosinophil genes, but they're actually genes that are encoded for by the esophagus itself, particularly the esophageal epithelium. And I want to be able to go into all of these, lucky for you, but I want to focus in particularly on some of these chief leads, like this one over here, which is called the 5q22 risk factor, where a gene called TSLP, a thymic stromal lymphopoietin, is sitting. Now, to give you an idea of how we can explore how this mechanistically works, we, with the support of the Cured Foundation, were able to generate mice that overexpress this gene. And what you can see here is that when you overexpress this, particularly in the skin of the esophagus and also the cutaneous skin, basically the squamous epithelium, you can see that the eosinophils accumulate in these transgenic mice that overexpress compared to the WT or wild-type mice. Now, the stain is an anti-eosinophil stain. So you can see a large amount of these staining cells, which are all the eosinophils in these mice that are overexpressing this gene product from the 5q22 locus. We also see on histological analysis under the microscope, and this is standard staining, that there's a change in the epithelium, certainly an accumulation of the cells, the eosinophils, a thickening of the scope of the wall of the esophagus, the epithelium, and in fact, take a look here. This is a microabscess with eosinophils accumulating, which we see in patients. And this is quantitating some of these changes. So the main point here is that if we overexpress some of these epithelial cell genes, which happens in patients, we can mimic the disease. Now, I'd like to now turn to a little bit of information about how the environment is likely to play a role. It turns out that the body is a host for environmental exposures, particularly bacteria. And the bacteria that we breathe and become colonized with normally lives in the GI tract. Now, we found a couple of years back that the esophagus has its own collection or community of bacteria, which we refer to as the microbiome. This is summarized in this particular slide, which is some of the critical findings that we have from our studies in both mice and humans. We now know that the esophagus has its own unique microbiome, dominated by a particular type of bacteria called Firmicutes, which includes Clostridium. It's unique from other positions in the body, including the mouth, the oral flora. The esophageal microbiome regulates the development of the epithelial cells in the esophagus. The microbiome, interestingly, can be reconstituted in the esophagus by a fecal transplant. And what that means is we can take stool, have the mice swallow it, and then we can convert a germ-free mouse into a normal esophageal microbiome with the stool itself. What that means is that the local environment in the esophagus dictates the community of bacteria that ultimately live there. And it also provides an opportunity for us to potentially treat patients through something that's emerging now, which is called fecal transplantation. Finally, what we found is that if we deplete these bacteria through antibiotic exposure or by looking at what we call germ-free mice, these mice are predisposed to EOE. They have heightened eosinophilic esophagitis, and they also have heightened or high levels of IgE antibody. In fact, if we look at patients with EOE, we can see that there's a dysregulation of the bacterial communities, a process called dysbiosis. And this is all influenced by environmental exposures such as antibiotics. So to summarize some of these points, we propose that the esophageal epithelium has a contributory role. And that's summarized in this diagram, where we now know that glucocorticoid treatments, which is swallowed, or we call topical delivery, can induce remission in EOE, which is an active biopsy on the right, converting to an oral biopsy on the left. The topical glucocorticoid bathes the epithelium of the esophagus. We also know that a fundamental problem in EOE is the loss of the barrier in the esophagus, which is the function of the epithelium. We know, as I showed you, that the EOE genes are in fact not eosinophil genes, but often genes that are expressed in the epithelium. Other work that we've done shows that epithelial proteases, which are enzymes, and antiproteases, which keep them in check, are disbalanced in EOE. And we also know that we can treat cells from the esophagus, particularly epithelium, with a cytokine called IL-13, and it will recapitulate some of the molecular features of EOE, which we refer to as the EOE transcriptome. So taken together, there's multiple lines of evidence that supports the epithelial cell hypothesis as the causal cell in this disease. Also want to draw your attention to mast cells. Mast cells, in fact, may have a contributory role in the disease. And the evidence for that was originally derived from our genetic work showing mast cell-specific gene expression in the esophagus. But also more recent work looking at something called single-cell sequencing analysis of esophageal biopsies. In this particular case, we isolate a biopsy, break it into individual cells, and then do genome sequencing on every cell at an individual level. And this allows us to identify nearly all the cells that are present in these biopsies. And what you can see here is a complex set of the epithelium, many different types of epithelial cells, different stages of the differentiation. What I want to draw your attention to is that there's a large population of mast cells that are resident in the esophagus and increased in EOE and actually activated. And this is a diagram that shows you some of the different genes that are expressed in different cells in the biopsy. And the more red the gene is, the higher the level of expression. And we can compare mast cells to myeloid cells, which are other white blood cells, B cells and T cells, which are other white blood cells in the biopsy, as well as other cells called fibroblasts, endothelium, and the epithelium itself. And what you can see is that the mast cells are very red because they express high amounts of cytokines and growth factors, different amounts of high levels of proteases, enzymes that code for lipid mediator metabolism enzymes and histamine producing genes as well. These mast cells also express different types of pleiotropic multifunctional receptors that allow them to respond to different environmental triggers, including the food directly. So this data strongly implicates mast cells. And in fact, just like we have a human eosinophil knockout, I'll come back to this at the end, there are ways now of creating mast cell human knockouts. And this is an approach currently being taken with antibodies against c-Kit antibody. And these studies are closing now. So early proof of concept studies will soon be emerging, testing the possible role of the mast cell in eliciting EOE. I want to just close by saying one of the other findings that we have, which is that the nerves may actually contribute to EOE as well. What we've recently found is that there's a series of sensory nerves that are running through the esophagus and they're dysregulated in the disease. Now, this is a picture of an esophagus. This is actually the wall of the esophagus. And it's a complex diagram, but if you give me some attention, you can actually become abstract a little bit thinking about what this is for those of you not used to looking at immunofluorescence staining. In this case, it's something called whole mount in situ staining, where the tissue has been dissolved, but we can see the fluorescent staining patterns of different cellular populations. First, I want to draw your attention to the blue stain, because that is this coding for a protein called E-coherent or epithelial cell coherent, which encodes for a protein that's expressed by the epithelium. So you can see that the epithelium runs longitudinally through this section. And this gives you an idea of where the lumen is, which is basically between the blue lines. Now what you can see is that red, which is certain type of sensory nerves, is running all along the lumen, right below the lumen and the epithelium. There's a whole bunch of sensory nerves that are present, and actually we can see them even penetrating into the epithelium. What you can also see here in white is that there is a network of autonomic nervous system, enteric nerves that run all around the esophagus. And these run primarily not longitudinally, but transversely, and they circumvent or go around the esophagus. Excuse me for not catching the right word, but you're getting the idea. They envelope the whole esophagus, and they form a web that's going all around it. But this is a beautiful image. But the main point is that the esophagus is embedded by nerves. And we think that these sensory nerves are very important in responding to the allergic triggers, but also in communicating with the brain. And this is illustrated further here, where we take a look at staining for tryptase, which is a mast cell stain in green. And you can see that this is a nerve cell in white. and that the mast cells that are degranulating are lying right next to the nerves. Similarly, eosinophils staying with eosinophil peroxidase are lying right next to these nerves as well. So the two cells are communicating, and that the inflammatory cells associated with this disease, eosinophils and mast cells, are likely activating the nerves. And in fact, the nerves may be activating the eosinophils. And this is a model that we have, which is early in its development, but we have strong evidence to indicate that the tissue where the allergic inflammation is releasing different cytokines that actually directly triggering nerves. In this case, the sensory nerves, including those that are embedded near the spinal cord, called the dorsal ganglia, respond directly to these inflammatory triggers, particularly IL-4 and IL-13. And this triggers pathways that activate inflammation in the brain, but also go back and feedback in the esophagus, causing a circuit that's bidirectional involving inflammation and the nerves. This is an early set of very important experiments that truly may tell us the complex contribution of the nerves to the disease process. And certainly one of our interests is to understand why patients have persistent symptoms such as pain, even in the absence of inflammation, which is likely to involve this pathway. So to put all this together, I propose that there's an integrated mechanism involving genes, environment, multiple cellular and cytokine signaling pathways. And this is a diagram that kind of illustrates that. But we know that there are genetic variants that predispose, particularly at 2p23 and 5q22. This leads to a predisposition to EOE, a liberation of pro-allergy cytokines, particularly TSLP. We can see the potent effects of TSLP that I showed you earlier. This targets the immune system, particularly dendritic cells and type 2 T cells that recognize food, and causes also mast cells to accumulate. And these cells release a cytokine called IL-13 that directly stimulates the gene product of 2p23 called calpain-14. It also downregulates proteins that are involved in barrier function. And importantly, IL-13 is a potent activator of production or transcription of the eotaxin gene, which is the eosinophil chemotactic factor and activating factor that elicits the eosinophil accumulation. Now, we know this is important for a number of reasons, but one is because there's very rare mutations that people can have, particularly in consanguineous families, that lead to loss of function of particularly the barrier genes, and this can lead to a nearly Mendelian inheritance pattern for EOE. Now, having defined this upstream pathway involving IL-4 and IL-13, this put us in a position to block these cytokines, and there's a number of ways of doing that. In our early work, we took an antibody approach called QAX576, which blocks IL-13. And we conducted at Cincinnati Children's Hospital a single-site, double-blind, placebo-controlled study testing the impact of anti-IL-13 on human EOE. And the important point is that we wanted to provide proof of principle, a proof of concept that this was an operational pathway. And indeed, we were able to do that through molecular analysis and certainly clinical observations. But I want to draw your attention to the approach that we took, which was a genome scan, looking at computational analysis of all the genes that were changing in the context of anti-IL-13 in humans in the esophagus. And this allowed us to find that IL-13 was upstream of all of these processes. These were fundamental processes involved in the disease, and this provided objective and definitive evidence that this was a pathway that was germane to this disease. And this, in fact, led to a number of different approaches, and obviously the first approval of a drug that blocks this pathway, dupilumab, for EOE. This is a very effective drug in most patients, blocking IL-4 and IL-13 and improving eosinophils accumulation, the barrier impairment, tissue remodeling, and other components as well, including all the clinical features that we can follow, such as dysphagia symptoms, endoscopic findings, and histological features. So this is a great advance in the field, and it shows the power of science in terms of bringing together new treatments. Notice that eosinophils are part of this, but they're not an essential part They're more of a ramification or a consequence itself rather than the primary defect. Our work has indicated that the esophagus is not just simple an organ that transports food, but in fact is an immunological organ. It has sensory nerves, it has its own microbiota, the bacteria, it has a tight barrier function. There's a large amount of proteases, antiproteases that are homeostatic. There's also evidence for another pathway which I didn't go in today, which is called the TGF-beta pathway, and its receptors that are involved. There's mast cells and other immune cells that are normally present in the esophagus, and there's also abundant expression of type 1, of IL-1 family cytokines that are often regulated at a high level because of their interaction with chromatin, and they can be released and trigger different responses and following triggers that are associated with diseases like EOE. So how about translating all of the findings into the future? Well, we know that we can block so many of these pathways with drugs, and this is very exciting and should be encouraging for patients to know that there's a pipeline of drugs. We've tried the eosinophil depleting antibodies. They're not having an impact in this disease, although they work for other allergies. We know that IL-4 and IL-13 blockade is very effective. We now have multiple trials of blocking TSLP, IL-33, mast cell depletion, as I mentioned, is soon to be revealed, the data there. There's ways of blocking the signaling, something called JAK inhibitors. There's also ways to restore the anti-protease and reduce the protease activity. I want to thank all the people that work together on this, the allergists that have worked together at the Cincinnati Center for Eosinophilic Diseases with GI doctors, the pathologists that are critical, research coordinators, the lab itself, other specialists, geneticists, special thanks to the Cured Foundation for hosting this webinar series this week and for supporting my lab in a very fundamental way. And I'm pleased and honored to call my lab the Rothenberg Cured Lab. Also want to thank the NIH for their long-term support. We know how important NIH funding is, and we want to bring that to your attention, especially as the government is considering modifications of NIH funding and structure. If you want to download any of this information that I showed you today, if you want to scan it to test your own hypotheses, use this VR code to get direct access to our data. We believe in sharing information. We have over 3,000 users that get access for free to the data that I showed you today, as well as others that are emerging. Thank you very much for your attention.